Chapter 3, Biological Foundations of Behavior. So in this chapter, we're going to be talking about the neural basis of behavior and the nervous system. Um, at first, the chapter looks a little intimidating. There's a lot of information in there. But if you break it down into two chunks, like I've done in this series of slides, there'll be two sections. There's the neural basis of behavior, where we're going to talk about neurons, and there's the nervous system. Okay, so it's two pieces. Yeah. So, in order to understand how the brain and neurotransmitters, how all of this affects our behavior, we first have to understand how neurons work. Okay, neurons are the tiniest piece. We have to understand about how they're constructed, the electrical activity of neurons, and how they communicate from one neuron to the next one and transmit information to different parts of the brain and the body. So to start this out, because there's a lot of detailed information that may seem overwhelming, okay, I sometimes like to start out with a short YouTube video where they give sort of a summary of everything in just a few minutes. And that just sort of gives us a base to go from, and then we can start at the beginning, okay?
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the neuron. This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA, or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
Okay, so that gives us a nice overview of what we're going to be talking about for the next 20 minutes or so. So let's start with the first part is the neuron structure. Like it was just explained in the video, it has three main parts. The dendrites on the far left, they receive messages from the neighboring neurons. The cell body contains the nucleus, which is the genetic information to determine how it develops and functions. Then the long piece there that the electrical impulse goes down is the axon. Okay, and it goes out to the end to the axon terminals, and then possibly onto the next neuron. We'll be covering the rest of some of the details there, like the myelin sheath, but we'll do that a little bit later. Okay. Another part that was mentioned in the text was about the blood-brain barrier. So just so you can see the difference, on the left there's a normal blood vessel, right? And that's found throughout the body. And then the one on the right is the brain blood vessel. okay? And it has this blood barrier that protects the brain from toxins. Toxins can enter the blood, go throughout the body, but the blood-brain barrier prevents any of those toxins from crossing over into the brain, and it does allow nutrients through. Okay, so that's the function of the blood-brain barrier. Another part is the glial cells. So the glial cells are found all around the neuron. Okay, it's from the Greek word for glue. So their job is to support and nourish and protect the neuron. So it surrounds them, it holds them in place. The glial cells also manufacture nutrient chemicals, absorbs toxins and waste materials. There are 6 different kinds of glial cells. We're not going into detail about the 6, but just to let you know that it's not just one type of cell that does this job. There's many different types of cells that perform this function. Okay, so those are the basics of the neuron and the structure, and now we'll move on to the electrical activity of neurons. So basically, neurons do 2 important things. They generate electricity, which travels down the axon, and they release chemicals, which is the form of communication. They release chemicals out the dendrites, which travel on to the next neuron. So nerve activation involves 3 basic steps. So when the neuron is at rest, the neuron has resting potential, as they call it. Okay, so due to the distribution of positively and negatively charged chemicals inside and out of the neuron, it's just sitting at rest. When it's stimulated, a flow of ions in and out through the cell membranes, it reverses the electrical charge of the resting potential, and it produces an action potential or a nerve impulse, which shoots down the axon towards the dendrites. Once that is finished and it's released out the end, the original distribution of ions is restored. The neuron is again at rest and capable again of being activated. Okay, so I have this is the picture from the text here. Okay, so you can see in the first one, there's the sodium ions and the sodium channel and the potassium channel. So the concentration of sodium ions outside the neuron and the negative protein ions inside contribute to the resting potential where it's just sitting. Okay, if the neuron is sufficiently stimulated, the sodium channel opens up and the sodium ions flow into the axon. So note that the potassium channels are still closed. So the sodium ions go rushing in. Sodium channels that were open in the third picture there, have now closed, and the potassium channels behind them are open, allowing the potassium ions to exit and restoring the resting potential at that point. Okay, so you can see there, it sort of shows the time in milliseconds about how long that takes. Now, I had said about the myelin sheath earlier. We were talking about the structure of the neuron. So as you can see, there's the dendrites, the cell body, and the axon comes down. And the myelin sheath, it's a fatty insulation layer derived from the glial cells, the glue that protects, right? So the job of the myelin sheath is to insulate the axon from the electrical activity and to increase the rate of transmission. So you can see there on the left is a healthy myelin sheath, which is totally coating the axon. Now, one example of what can happen if the myelin sheath is damaged, you have the exposed fibers and you ended up with nerve damage, and one example of that is multiple sclerosis. And this is caused by the immune system attacking the myelin sheath. Okay, so the second part is the communication in the synapse. Okay, so synaptic transmission. Neurons don't make actual physical contact, right? So the neurons are not touching each other. They communicate with chemicals. So the space that's between the two axons is called the synaptic cleft. Okay. So when the impulse comes down the axon and it comes down to the end, right? It's going to release a neurotransmitter into the synaptic cleft, which is going to travel to the next axon, right? So they bind to the receptor sites at the postsynaptic neuron, which is the next one. They only bind to certain ones. So they'll bind there and then they'll enter, and then the electrical impulse or neurotransmitter continues down the dendrites of that neuron and continue on to the next. I hope that makes sense. So here's another picture of how that works, right? So you can see there the nerve impulse. It comes down. You've got your synaptic vessels there. They shoot down towards the synaptic cleft and drop the little red circles, right, into the space there, and they get absorbed into the next neuron. And like I said, they only fit into certain receptors, right? So serotonin is going to come down and it's going to be released into the synaptic cleft, and it is only going to be picked up by serotonin receptors, right? It's like a puzzle. Each piece fits, like you can see in the upper right-hand corner there. So transmitter will fit in the receptor, yeah, according to the shape, which is okay. So when the neurotransmitter goes down, it has, and it goes into the next neuron. It's going to do one of two things. Either it's an excitation neuron or it's an inhibition neuron, right? So an excitation of the postsynaptic neuron, it depolarizes the neuron. It stimulates the flow of the Na ions, and it increases the likelihood of action potential. So it's getting excited, it's firing it up. Okay, or what it can do is have an inhibitory effect and deactivate. So it hyperpolarizes the membrane, makes it more negative, and it decreases the likelihood of an action potential. There's a YouTube video link there if you wanted to look at that a little bit further. I always find them helpful, even to just watch them over and over again, just so that the words start to sink in and really feel natural, and get to know all the terms. Okay, so I'm putting this video in here, okay, because I think it's helpful. Please excuse the slightly monotone voice of this gentleman, but it is a good explanation of how this works. Okay.
Greetings, psychologists. I'm just going to provide a brief and broad overview of the distinction between the excitatory and inhibitory effects of different types of neurotransmitters. So first, an overview. So here we've got a presynaptic neuron, and as a result of an action potential, it's started in the cell body and conveyed along the axon to the axon terminals, where we've got some vesicles that are storing the neurotransmitters. Those neurotransmitters are going to be released into the synapse. Some of these are going to bind with the receptor sites on the post postsynaptic neuron. More about that in a future clip. And that's going to have one of two effects, either an excitatory or an inhibitory effect, which is the purpose of this video. So let's zoom in and have a look at excitatory neurotransmitters such as glutamate. So here we've got the cell membrane. So on this side we've got inside the postsynaptic neuron, and here we've got the outside. And what you'll notice on the outside of the postsynaptic neuron is we've got a bunch of sodium ions. These are positively charged. And when a excitatory neurotransmitter such as glutamate binds with the appropriate receptor on the postsynaptic neuron, it'll briefly open up these channels. And what's going to happen is we're going to get a bunch of this positively charged sodium that's going to go from outside the cell membrane to inside. So therefore, the effect of this is that it's going to depolarize that postsynaptic neuron. It's going to make it less negative, and it's going to have an excitatory effect, which means it's going to be more likely to fire. And so as a result of that, glutamate is going to be pulsating throughout the central nervous system, and it's going to play a key role in cognition, learning, and memory in particular, which we'll look at later in the course. Inhibitory neurotransmitters, on the other hand, are going to have an opposite effect. So in this case, we've got a bunch of potassium ions, which are located, the K1s here, located on the inside of the postsynaptic neuron. And when an inhibitory neurotransmitter binds with the receptor sites on the postsynaptic neuron, it's going to open up some of these potassium channels, which we've got right here. And some of this potassium, which is positively charged, is going to leak out of the postsynaptic neuron. And another thing that can happen is we can get some negatively charged chlorine ions that can actually come in to the postsynaptic neuron. And so what's going to happen is the postsynaptic neuron is actually going to be hyperpolarized, or it's going to become more negative, and it's going to be less likely to fire. And you might say, what's the importance of that? Why does this occur? Well, GABA, which we're going to learn a lot about in Unit 4 in particular, when we look at some mental health issues, particularly with phobias, it counterbalances the effects of glutamate. So it has a calming effect. So if we didn't have inhibitory neurotransmitters like GABA and serotonin, your brain would be overly wired. We'd be highly anxious. It'd be hard to switch off. So GABA plays a very important role in counteracting the effects of excitatory neurotransmitters such as glutamate, so that we have an optimal level of functioning. And when this becomes out of balance, which we're going to learn about later on, that's when we can have various disorders such as anxiety disorders and phobias.
Okay. Like I said, a little monotone, but some good information there. And there's just a chart that sort of summarizes that, which I don't need to go over again. Okay, so once they've activated or inhibited a neuron, so what happens to the neurotransmitters then, right? They do one of two things. They either break down, so other chemicals, they're in the cleft there, the red dots that are in the cleft there. Okay, they're not binding, they're sort of drifting around there, but it's the action's been completed, right? So they'll either break down, so other chemicals in that synapse will break them down into their chemical components and they disappear. Or there's a thing called reuptake. Okay, and this is an important one. Reuptake means, you see those two little red ones that are sneaking back into the yellow neuron? Okay, that's the neurotransmitters being reabsorbed into the presynaptic axon terminal. So they get absorbed back into where they came from, and that's called reuptake. Okay? So we've kind of reached the end here. Okay? This is a video, and I'm not going to play it right now. You can go back through these slides or just this one. And this is another video that goes through each of the different pieces. I tried to find some different videos that are going to go over all the difficult little sections that you can rewatch and rewatch. Okay, so this is a summary of everything that we've talked about in this video here. Okay, and that brings us back to the end of the neural basis of behavior. We talked about neurons, we talked about their three different parts, we talked about the electrical activity and how the potential in the electricity travels down the axon and how it crosses over, and we also talked about how neurons communicate, okay, from one neuron to the next by the neurotransmitters crossing over in the synaptic cleft. Okay, so that is the end of the first section. The next section is the nervous system, and I will be discussing that in the next video.